Welcome friends, welcome to the lectures of biology, and today we are going to discuss about the chapter on principles of inheritance and variation. So let's start the chapter. Now the most important term to introduce this chapter is the definition of genetics. Genetics is the science which studies the principles of inheritance and variation. Now, what do these two terminologies, inheritance and variation, mean? Inheritance means the process through which the characters are passed from parents to the offspring. So, it is the study of the process. What is the variation? Variation is the degree with which the offsprings differ from their parent, or they differ from or among themselves. That means the offsprings of same parents may differ from each other. That accounts for the variation. Now in the primitive times, before the scientific phenomena which were discovered for explanation of such processes, humans have already used this variation, and they knew how sexual reproduction introduced different types of variations in the upcoming progenies. Previous populations or the study of different types of domesticated animals and the plants, their varieties, this has been already done by the previous people, and it is through artificial hybridization or cross pollination. And for that we have the example of Sahiwal cow of Punjab, which have been derived from the wild relatives of these cows. Now coming to the Mendel, who was the person behind all the science which involves inheritance and variation. So Mendel basically performed the hybridization experiments on pea plants, the scientific name of which is Pisum sativum. He included the study of 7 contrasting characters. The characters which were studied by Mendel were stem height, then flower color, flower position, pod color, pod shape, seed color, and seed shape. So these 7 characters were studied by Mendel one by one, each at a time. Now these characters have their contrasting traits. That means each character has 2 variations. For stem height we have tall and dwarf. For flower color we have violet and white. For flower position we have axial and terminal. For pod color we have green and yellow. For pod shape we have inflated and constricted. For seed color we have yellow, green, and finally for seed shape we have round and wrinkled as contrasting traits. Now Mendel selected the plants of pea which contained one character differing. The difference was between one character, and rest of the character between these 2 varieties were same. So he included in his hybridization experiments total of 14 plant varieties, 7 for male partner and the other 7 for the female partner. Now let's discuss the hybridization experiments performed by Mendel. So what did he do? He first of all selected the plants for the character stem height. Now as we know that this stem height has two contrasting traits, that is tall and dwarf, he produced two pure line of the tall plants and the dwarf plants. Now what is this pure line? Pure line or pure breeding varieties are those varieties which breed true to their character. Now these pure lines breed true, what does it mean? It means that these pure line varieties will give the progenies which will resemble themselves in the character as it was present in the parents. So these pure line will give same character in the next generation. That means they are stable for the inheritance of the character for which they are chosen. So suppose here is for stem height. He took tall plant and dwarf plant. Now this tallness, if the gene responsible for this tallness is to be represented by alphabets, we can represent it by capital T, capital T. And for dwarfness, we can represent by small t and small t. Now when Mendel hybridized these two types of plants, he obtained all tall plants in the next generation, that was known to be F1 generation. After obtaining all tall plants in F1 generation, Mendel conducted the selfing or self-pollination of these plants of F1 generation. After selfing these plants, they produced F2 generation, and in F2 generation, Mendel obtained tall and dwarf plants in the ratio of 3/4 is to 1/4. That means the ratio obtained by Mendel in the F2 generation was tall for 3/4 and dwarf for 1/4. Now what did he observe by these experimental results? First of all, he observed that in the first generation or first filial generation, there was presence of only a single character, and that character was dominating the other characters. So he devised the law of dominance from this particular result or analysis. Now next, in the F2 generation, he found out that although this dwarfness was not available in the F1 generation, but this character reappeared in the F2 generation. Now what does it mean? It means that this character was not mixed with the tallness, that there was not any kind of blending of the characters, therefore only this character reappeared. So from here he got an idea to devise a law which is now called as law of segregation. We will be dealing with these laws in a bit detail, but for the time being, how the Mendel conducted the experiment and how he analyzed the result, this you have to understand. Okay? Now let's move to the fact that when Mendel involved only one character at a time, such cross was called as monohybrid cross. So what is a monohybrid cross? Monohybrid cross is the cross or hybridization which involves one character at a time. Sorry. Now before studying into or going into the details, let us understand some terminologies related to genetics. First terminology is factors. When Mendel analyzed the results of the experiment, hybridization experiment he conducted, he found that there has to be some mechanism or something which is transmitting the character of tallness or dwarfness from one generation to next generation to further next generation. That particular thing or whatever thing he imagined was termed by him as factors. The factors were responsible for the transmission of characters from one generation to the other generation, and now these factors are known to be genes. Next point, in a diploid individual, these factors are present in pairs, and these pairs or the members of these pair are called as alleles. Now alleles, what are they? They are basically the genes or the factors devised by Mendel, but they are slightly different form of same gene. You can say slightly different or alternative form of the same gene. That means, what is the meaning of same gene? The gene which is controlling the same character, here in this case the plant height. So now we have got the definition of alleles. Now there is a condition which is known as homozygous or homozygosity. Homozygous condition is that when both the alleles are similar, that is capital T or small t small t. There is another condition which is known as heterozygous condition. In this condition, both the alleles are different, and here is the case of heterozygosity. So in this manner, Mendel could identify or could devise a mechanism to know the genotype of the pea plant, of which type of genotype, whether homozygous or heterozygous. Now let us again very briefly discuss the inheritance pattern of the monohybrid cross conducted by Mendel. For tall plant, pure tall, this is pure dwarf crossing, it results into formation of a heterozygote, it will also be tall. Now in the F2 generation, we have capital T small t and small t. These three types of plants. Now these three types of plants are based on the genotype, because their genetic makeup is different from each other. Now we can analyze this result with the help of Punnett square. What is Punnett square? Punnett square is a checkerboard arrangement which is used to calculate the probability or possibility of all types of genotypes in upcoming generation. So now what we can do here, this is the plant of F1 generation. This plant will produce gametes, and these gametes will be of two types, capital T and small t. Now as selfing of this plant was conducted, so we will get together total 4 gametes. Now very simple, we can hybridize and ultimately we get this status. Now there is one capital T, there are two capital T small t and one small t. So this ratio of a monohybrid cross in F2 generation is known as genotypic ratio. Now if we look at the morphology or phenotype of the plant, what is phenotype? It is the external appearance of the plant. So if we see the morphology of the plant, both these two types of plants will be tall and this will be dwarf. So if we are to observe the phenotypic ratio, then tall is to dwarf will be 2 plus 1, that is 3 is to 1. This will be called as phenotypic ratio. So these are very important, phenotypic ratio and genotypic ratio. Now let's move to the test cross. Suppose after hybridization experiment, Mendel got a tall plant. In morphology or in appearance, it is tall. How will we get to know that this tallness is because of capital T or because of capital T small t? So for this purpose, a test cross is performed. What is a test cross? A test cross is the cross of an individual with dominant character with its recessive parent, that is dominant individual is crossed with recessive parent. For test cross, we can take the example of flower color. So we see that flower color is of two types in pea plants. One is violet, and this violet is dominant, and the other is white, which is recessive. The genotype of both being capital W and small w, small w. Now we have to perform a test cross in which a dominant individual is needed, and we do not know the genotype of the dominant individual. So it can be either capital W or capital W, small w. Now if we cross this genotype individual with recessive parent, that is small w, what do we get? We get two types of gametes from this parent, capital small w, and from this type of gamete also, capital W. We can write it here, no problem. This will be capital W, capital W, capital W, small w, and same here. Now we see that all the progeny, they have same genotype as capital W, small w. That means all will be violet in color. The flowers of all the progeny will be violet in color. Now let's move to the next possibility. This is crossed with the recessive parent small w. So that possibility we can draw here. The gametes are made as capital W, small w. Now after hybridization, we get two types of individual, that is capital W, small w, and small w, and both these two types of individuals, they are found 50-50, that is ratio will be 1:1. So now if the ratio of the cross, the test cross, is 1:1 for one character, that means the dominant individual will be heterozygous, and if this ratio or all the individuals coming in the next generation, they are having the dominant phenotype, the condition or genotype of the dominant individual will be capital W, that is homozygous condition. Now in this manner we come to know that through monohybrid experiment or monohybrid cross, Mendel came to give two sets of generalizations, and those generalizations are now called as the Mendels Law. And these two laws of Mendel, which are based on monohybrid cross, are law of dominance, and second is the law of segregation. Now, if we have to define the law of dominance, it is defined in three different steps. First, that the factors controlling a character are present in a pair. That means factors are in pairs. Okay? Now, factors are in pairs, but from where the factors have come, this has to be defined. That means the definition prior to this should be characters are controlled by factors. Okay? So, first definition should be that the characters are controlled by factors or genes, and the second definition should be the factors are present in pairs. Now, what is the third definition or third part of the definition of law of dominance? It says that in a heterozygous individual, one of the factors dominates, which is called as dominant factor, over the other, which is called as recessive factor. So, heterozygous contain two types of factors, or we can say two types of alleles, allele 1 and allele 2. Now, this allele 1 will be dominant and the allele 2 will be recessive. This is the typical part of the definition of the law of dominance. Now, what is the law of segregation? The law of segregation states that during the formation of gametes, yani ki, that means during gamete formation, the factors present in a pair, they separate or segregate from each other, and ultimately they fuse during the time of fertilization. So, during gamete formation, factors of a pair separate or segregate, which later fuse during fertilization. Okay? We have already witnessed this phenomena. How? Suppose this was a tall individual and this was the dwarf individual. As this is a diploid one, it will contain two factors. This will also contain two factors. Now, both these factors will separate from each other during the gamete formation. So, one gamete will be formed like this and the other will be formed like this. Similarly here, small t and small t. This is the phenomena which defines the law of segregation, and afterwards they will fuse with each other and the diploid condition is again restored. So, these laws, they were based on the monohybrid cross performed by the Mendel. Now, we will deal about the exceptions of the monohybrid cross in the next video. Do continue to watch and learn biology. Thanks a lot.